Dear learners, today we will discuss about nucleic acids. Nucleic acids are long-chain polymeric molecules. The monomer, that is the repeating unit, is known as the nucleotides, and hence sometimes nucleic acids are referred to as polynucleotides. Deoxyribonucleic acids and ribonucleic acids are two major types of nucleic acids. DNA and RNA are responsible for the inheritance and the transmission of specific characteristics from one generation to another generation. The nucleic acids function. Nucleic acids are responsible for the transmission of inherited characters from always the parents to offspring. They are responsible for the synthesis of protein in our body. That is, it's nothing but that is the central dogma. First, initially they are the productions of daughter DNA from parent DNA to daughter DNA to RNA, finally productions of protein. So final the productions of protein it needs for nucleic acids. Apart from this, responsible for the synthesis of protein, it also helpful for the DNA fingerprinting is a method used by forensic expert to determine paternity. It is also used for the identification of criminals. It also played a major role in studies regarding biological evaluation and genetics. The classification of nucleic acids, nucleotides, nucleosides, and phosphoric acid. First, the nucleic acid is nothing but that is it consists of polynucleotide. Polynucleotide is nothing but the combinations of nucleosides as well as the phosphoric acid. The nucleosides it's nothing but that is a nitrogenous bases and sugar molecule. The sugar molecule first that is they are the two part. One is the ribose sugar as well as the deoxyribose sugar. Nitrogenous bases purine and pyrimidine. The purine bases always the adenine, guanine. The pyrimidine bases cytosine, uracil, and thymine. Instead of uracil, they may be present in the thymine. It's called as a DNA. If they present in the uracil, means it's a RNA. Only one part is the difference. This structure show that the purine structures as well as the pyrimidine structure. Pyrimidine structures cytosine, uracil, and thymine. Purine, adenine, as well as the guanine. This structure show that the DNA as well as the RNA. RNA is nothing but ribonucleic acid. DNA, deoxyribonucleic acid. DNA or RNA, both of them they consist of the nucleotides. Especially for already you know that is a nucleotide is nothing but nucleosides as well as the sugar molecule. So nitrogenous bases, purine as well as pyrimidine bases, that is adenine, guanine, cytosine, thymine. It's called as a DNA. Adenine, guanine, cytosine, uracil means it's a RNA. So the DNA it is a double helical model of DNA. The RNA it is a single stranded of RNA molecules. Nucleosides. The nucleoside is structural subunits of nucleic acids, the heredity controlling components of all living cells consisting of a molecule of sugar linked to a nitrogen containing organic ring compound. In the most important nucleosides, the sugar is either the ribose as well as the or deoxyribose. The based upon the presence of ribose as well as the deoxyribose sugar only they are conformed with the DNA or RNA. Cytosine, thymine, uracil, guanine. Cytosine, thymine, uracil. It's called as a pyrimidine nitrogenous bases, purine, adenine, as well as a guanine. This structure show that the nucleosides. Next one, nucleotide. Nucleosides as well as the sugar molecule. Like a nucleotide is nothing but it is an organic molecule with a basic composition of a nitrogenous base, pentose sugar, and as well as a phosphate. The DNA and RNA are polynucleotides which contain a chain of nucleotides monomer with different nitrogenous bases. Nucleotides are essential for carrying out metabolic and physiological activities. ATP, adenosine triphosphate. Based upon the phosphate group, they are called adenosine triphosphate. It acts as always the energy currency of cells. The nucleotides form various coenzymes as well as the cofactors like NAD, NADP, FAD, coenzyme A. The structure, a nucleotide, it consists of three units. One is a nitrogenous basis, that is a purine and pyrimidine basis. Second one is a pentose sugar, it's like a ribose as well as a deoxyribose. Phosphates, that is the based upon the number of the phosphate group will be present. It like maybe only one phosphate group, it's called as a monophosphate. The two phosphate group, it's called as a diphosphate, triphosphate. This structure show that the nucleotide, that is a nitrogenous basis as well as the phosphate group and sugar. If they may be the ribose sugar means it's called as a RNA. They may be deoxyribose sugar means they may be present in the deoxyribose sugar. Next we'll move on to the double helical model of DNA. The double helical model of DNA, it's otherwise called as a Watson and Crick model of DNA or structure of DNA. A nucleic acid is a polymer of a nucleotide monomer. In other words, it may be considered as polynucleotide. Already you know that polynucleotide is nothing but nucleosides as well as the sugar molecule. The polynucleotide, that is a deoxyribonucleic acid base deoxyribose phosphate nucleotides units are joined together by 3' to 5' phosphodiester bonds. The sequence of specific bases is termed as the primary structure. The purine and pyrimidine bases of DNA carry genetic informations, whereas the sugar and phosphate groups perform a structural role. Watson and Crick model of DNA or Watson and Crick double helical structures of DNA. James D. Watson of United States, Francis H. C. Crick of England in 1953 proposed the now familiar 3D model of DNA. For this discovery, where they are awarded the prestigious Nobel Prize in 1962. Some of the silent features of Watson and Crick model of DNA. The DNA molecule consists of two long polynucleotide chains which are wrapped around each other, the form of right-handed double helix. The two chains are joined together by specific hydrogen bond. The diameter of the helix is 20 Armstrong. The distance between each term of the helix is 34 Armstrong. The bases are 3.4 Armstrong apart along the helix axis. Each turn of the helix contains 10 nucleotides. The twistings of the strands results in the formation of major grooves as well as a minor groove. The two strands run in opposite direction. They are anti-parallel. One strand run in the 5' to 3' direction and the other strand run in 3' to 5' directions. In DNA helix, the hydrophilic deoxyribose phosphate backbone is outside of the molecule, whereas the hydrophobic bases are stacked inside the perpendicular to the helix axis. The two chains held together by hydrogen bonds between pairs of bases. Every base pair consists of purine from one strand and pyrimidine from other strand. Adenine always pair with thymine with the two hydrogen bonds. Guanine always pair with cytosine with 3 hydrogen bonds. It's called as a Chargaff rule. The Chargaff rule, based upon the Chargaff rule, the bases adenine always pair with thymine by the 2 hydrogen bond. Guanine always pair with the cytosine by the 3 hydrogen bonds. The biological significance of DNA. It is responsible to carry information from generation to generation. It is the most important stable molecule. It serves as a template for RNA synthesis. It contains a genetic code which guide the process of protein synthesis in cells. It regulates all biological activities of cells. Next, we'll move on to RNA structure, occurrence, types, chemistry, and biological functions. What is the RNA? RNA is nothing but RNA is a ribonucleic acid that helps in the synthesis of proteins in our body. The nucleic acid is responsible for the productions of new cells in the human body. It is usually obtained from the DNA molecule, like a transcription process. Based upon the presence of sugar molecule, ribose sugar, as well as the transcription process, they are productions of RNA molecule. The RNA, it's finally named ribonucleic acids. These are the structure. The RNA, ribonucleic acid, ribose sugar, nitrogenous bases, adenine, guanine, cytosine, as well as the uracil. It consists of, they formed to helical shape. Basic structures of RNA. The ribonucleic acid has all the components same to that of the DNA, with only two main differences within it. RNA has the same nitrogen bases called adenine, guanine, cytosine, except the thymine. The thymine is replaced by uracil. Adenine and uracil are considered as the major building blocks of RNA, and both of them form base pair with the help of two hydrogen bonds. RNA resembles a hairpin structures and like the nucleotides in DNA. Functions of RNA. RNA involved in a variety of functions within the cells and are found in all living organisms, including bacteria, viruses, plants, and animals. RNA serves as a structural molecule in cell organelles and also involved in the catalysis of biochemical reactions. The primary functions of RNA, it facilitates the translation of DNA into protein, functions as an adapter molecule in protein synthesis. It serves as a messenger between the DNA and a ribosome. They are the carrier of genetic information in all living cells. It promotes the ribosome to choose the right amino acid which is required in building up of new protein in the body. tRNA, it's nothing but transfer RNA. It is also one of the type of RNA. It helps in the synthesis of protein from mRNA. tRNA function as an adapter molecule during the translation process. As an adapter, it links the amino acid to nucleic acids. It carries the amino acids to be added in the peptide chain and also disappears the codon for the same in the mRNA molecule. Structure of tRNA. Generally, 76 to 90 nucleotides long of RNA molecule will be present in the tRNA. Each amino acid has specific tRNAs. The amino acids of the codons always having the starting codon as well as the stop codon. Start codon is nothing but AUG, stop codon UAA, UAG, UGA. The stop codons are not recognized by any tRNAs. The secondary structures of tRNA look like a clover leaf. The folded structure is formed due to hydrogen bonding between the complementary bases. What is the main functions of the tRNA? The tRNA plays an important role in protein synthesis. It acts as adapter molecule for linking amino acids to its specific codon present in mRNA. Amino acylation of tRNA is the first step in protein synthesis. The protein synthesis is the process of translation process in the ribosome subunits. The amino acids to the growing polypeptide chain in the ribosomes. This decoding of codons of mRNA by specific tRNAs continues until the entire sequences for a polypeptide chain is translated. Next, mRNAs. Messenger RNA or single stranded molecule in cells that transfer genetic information from the DNA in the nucleus to the cytoplasm, where protein are synthesized in the ribosome. mRNA are a group of RNAs that can be translated into proteins, while other RNAs there is no involved to the productions of protein in the ribosomes. In prokaryotes, mRNA contain a transcript copy of the DNA sequences with a terminal 5' triphosphate group and 3' hydroxyl residue. In eukaryotes, the structure of mRNA molecule is more elaborate. The 5' triphosphate residue is further esterified, forming a cap structure. The 3' ends typically contain in a poly A tail that consists of multiple adenosine monophosphates and is added enzymatically after transcription. In general, due to the lack of the poly A tail and the cap structures, prokaryotic mRNA are less stable than eukaryotic mRNA and degrade very rapidly. These are the mRNA structures. The one side it's having the 5' cap in the presence of phosphate group, and another side 3' that is having the A basis poly A tail, and also the having the coding region. One is first the coding region also having the starting site as well as the stop codon site. Starting site U starting site AUG, stop codon site UAA, UAG, UGA. mRNA function, always the mRNA it involved to the genetic information from DNA to RNA and to protein like a central dogma. Because DNA cannot be decoded directly to produce amino acid, so it needs for RNA. mRNAs act as an intermediary between the DNA and protein molecule. Each piece of mRNA encodes the information for one protein in eukaryotes. However, the protein is the production from the amino acids. Every three nitrogen containing bases like AUG. AUG it's one of the codon. It will be helpful for the productions of one type of amino acid. All the amino acids join to form a protein molecule. So the main role, intermediary step between the DNA and protein. So mRNA is the main role for the productions of protein molecule. So initiation, elongation, termination, all the process it occurs in the translation process. rRNA, the RNA found in ribosome, the molecule responsible for catalyzing protein synthesis is known as a ribonucleic acid. Over 60 to 80% of the weight of the ribosome is composed of ribosomal RNA, essential for all of the ribosome's activities, including binding to mRNA, attracting tRNA, and catalyzing the formation of peptide bond between amino acids. These are the rRNA structure. They having the two type of the subunits. One is the large subunit and small subunit. Types of rRNA in prokaryotes. In prokaryotes, the 16S ribosomal RNA is housed in a compact 30S ribosomal subunit. The two rRNA species are present in the large 50S ribosomal subunit, 23S as well as 5S ribosomal rRNAs. So it consists of the rRNA types in the prokaryotes, 16S, 23S and 5S rRNA. In eukaryotes, it consists of the 60S as well as a 40S subunit. Usually have several varieties of the rRNA genes arranged in repetitive sequences, like the 60S subunit contains 28S rRNA, 5.8S rRNA, 5S rRNA, whereas 40S subunit contains 18S rRNA. The functions of rRNA: protein synthesis is the primary functions of rRNA. By attaching to messenger RNA and transfer rRNA, this molecule assure that the codon sequences of the mRNA is approximately translated into the amino acid sequences of protein. Finally, the difference between DNA and RNA. DNA sugar moiety in deoxyribose. The RNA, the sugar moiety is ribose. The bases present in the DNA, adenine, guanine, cytosine, thymine. RNA, adenine, guanine, cytosine, uracil. The DNA molecule always the double-stranded molecules. RNA, it is a single-stranded molecule. It is DNA, it will obey the Chargaff rule. Already you know that the Chargaff rule is nothing but adenine always pair with the thymine, guanine always pair with the cytosine. But the RNA is also having the Chargaff rule, but it does not obey the Chargaff rule. Bases are not modified. Bases are modified in RNA. It is stable and not hydrolyzed easily by alkalis. It is unstable, hydrolyzed easily by alkalis. DNA content in constant in all the cells except during cell division. It varies from cell to cell. The lifetime of DNA is comparatively high. RNA is a very short-lived. No natural DNA is catalytic. RNA can be catalytic. It present in the nucleus, mitochondria and chloroplast. It's a DNA. The RNA is always present in the mitochondria, nucleus, nucleolus, ribosome.